go for it. All right, so we left off with an intro towards where we're headed with inheritance. We found out that traits don't just blend together randomly. Some traits can mask the other, some can hide, things like that. Today is mainly going to focus on mitosis right here. And I really like using the zygote, the first cell that comes at the end of two organisms combining to form an offspring, because although today's focus is going to be mitosis, I like seeing kind of where these two can link to each other. Most of the exam questions will be like, there will be a sort of a compare contrast between meiosis and mitosis. That's how I like to test you on this. It's much better than being like, what's anaphase? You know, it's kind of boring. Plus, you got the two sides of notes, so I can ask you something a little bigger. But in humans, and that's kind of noted right here, this is the basic equation. You got 23 chromosomes. Dad divides those in half, mom divides those in half, they recombine, and then you end up as the zygote, this first initial cell that everybody in this room eventually started as that single one. Now from here, everything flows through mitosis. You need to make more of these. And mitosis is a very good way of cleanly and accurately copying the cell. Not just the cell components, but importantly, and we'll follow, mitosis is a lot easier to follow if you just follow the DNA. And this is a DNA class, right? This isn't cell bio. So that's the emphasis. Now, this was a big question, again, back in the day, before we have any idea of what's happening. How in the world can one cell become 10 trillion like we are? How can that accuracy be maintained? How can that integrity be maintained so well? So the key points of this class, as far as cells go, there are some pieces of unified cell theory that are more important in this class than others, mainly that we're going to need to replicate the information. But two key points, obviously, here with cells. They're the basic unit of a living thing that we can, with certainty, call a living thing. I'm always happy to debate if viruses are alive. I'm always even happier to debate if a cancer or tumor population is alive, but that's a whole nother argument. Now, the other key point too, and that's basically just all we're going to see today with mitosis, is cells come from other cells. Easy enough. Now, the good thing here is like, don't have to worry about any Krebs cycle stuff. Scary. We don't have to worry about too much cell function today. Obviously, we can get into how information and replication can change across mitosis and how that factors into evolution. But for the most part, mitosis is and has evolved to become a very clean, nice process. You don't want a ton of inconsistency when you're copying cells, right? All the cells in your body, like we talked about, they have the same genome. So the full genome is going to start in the first zygote, like we talked about, and it will be copied to all trillions or millions of cells that become the organism, the whole thing. It's gigantic. Like our genome, like I said, about 4.6 billion nucleotides. You're going to copy that whole thing every time. We'll see later in the class how that's made possible. But this is a key thing. Every single cell has the same set of instructions. It comes with the same book. Each cell can decide what pages to read to become different cells, but they've got all the same information. And so in the beginning down here, this little like mollusk right here, this was the first organism that we could use under like a very not great working microscope. But you can start to see how, but people started to observe that 1 would become 2. And that 2 could become 4, so on and so forth. Now, as we sit here now, our cells are not exponentially doubling all the time. We'll get later on today, we'll see how that process stops, controls itself, and moves on. But cool thing, this was only in 2000. We only did sequence the entire human genome about 20 years ago. And we have the whole pile, all 4.6 billion nucleotides. Pretty cool. It's a fun story, and we'll get into it later. One key element of the story that, although today it's free to scroll, everything's okay, the genome was almost made a subscription service. Yeah, so the guy up here, Baldy, Solera, Craig Venter, he was like, I'll only help you if you give me the top 100 most needed medical genes, and people to study them will have to pay me an honorarium every year. Now, luckily, we apparently talked him down from that, but yeah, wouldn't have been a great time, to say the least. But today, you can scroll through this entire thing, and our cells have to copy that every single time, this gigantic thing, right? Each little gene located on one specific area of a chromosome, everything needs to get doubled each time. Now that means, and I don't think we're quite here yet, but as a single cell, this is how your single cell is going to look right here. You have one copy of mom chromosome number one copy of dad chromosome number 1, for example, dad, mom. So most of your cells exist like this. Now, to double them, in that S phase of mitosis that you remember from intro bio, you'll actually double this amount right here. And that double will be a clean carbon copy of everything. Keep that in mind. Because obviously, one cell becoming 2 cells means you need 2 genomes all of a sudden, and they need to be identical as possible. Okay. Pretty basic here, but we have yet to find any source of life that doesn't store information as DNA. DNA, its function is not to go out and do stuff. Its function is that it's very safe. It's a very stable chemical, okay? Let's just go here. It's a very stable chemical. Super safe. Super easy to store. Everything's all locked up in a nucleus. It's got 2 layers around it with a cell membrane, obviously. So all the pieces that you need to make any organism, from a banana to a human to a plant. Wait, bananas are plants, sorry. They all use the same code and the same thing. Now, we'll get into these later, histones, all this stuff. Don't worry about this. This is blue text for now. But there are ways that we can store this massive amount of chemical in every cell and compact it. But ultimately, DNA is a great way to store all this info. It's very safe. DNA left out even at room temperature in a tube, it's not going to degrade for years. It's great. Okay, so we've seen this before, but this is a little deeper look at that more cartoonish slide we had last time. The role of DNA is to store stuff. DNA stays nice and safe in the nucleus. Like I said, if you needed to make a copy out of the book of information that you have, you don't just rip the page out. You copy the page down into RNA, and that's going to get transported out of the safety of the nucleus. So RNA's role is simply to transport and copy the information from the genome. Now, the reason behind that, too, is that the cytoplasm of a cell is a very dangerous place. You got a lot of enzymes, you got a lot of damaging agents. RNA does not have a long lifetime. It's very fleeting. Now, in that fleeting life, RNA's entire role is to become a protein, or is at least to transmit that information into that protein language. And don't worry if this feels like review from yesterday, it is. It's okay. Just want to make sure this is hammered in. And as I kind of emphasized yesterday, proteins pull off the functions in your cells. The receptors, the enzymes, the defense mechanisms, the borders, everything. They're what do stuff. DNA and RNA, very limited role in doing anything. Protein does it all. Amino acids are highly diverse, highly capable of almost infinite function. Now, doesn't mean the DNA is not important. Doesn't mean that if you change the DNA up there, you're going to change the RNA, which means you're going to change the protein. Some of these changes are manifested as something called alleles. So if you break down each level, oops, we have 23 of these chromosomes, right? And in this case, they are kind of pushed together like this in this formation. Across these 23 chromosomes, you got about 22,000 genes or so. You don't need to know that number. I'm just trying to give you the scale here. Now, like we're highlighting kind of over here, the gene will be on the same spot for mom and dad. But they can be slightly different versions of the same gene. And by a slight change, if there's a slight change in the DNA that causes a, let's say, slight or major change in the protein, right? Remember, the protein does stuff all of a sudden. If you make that slight change in a gene between these two, you can have a wildly different result. Now, alleles are still, they're in the same location. The allele for brown eyes and the allele for blue eyes, they're the same gene, just with a couple differences. They're in the same spot on chromosome, I think, 3. So they never move or anything. Alleles don't move. You just happen to get an allele from mom and an allele from dad that happen to be different. And plenty of time you can just have 2 of the same alleles, and that's it. One from mom, one from dad. Now, some genes come with just one version of themselves. Can't change it. Other genes come with a recessive dominant, kind of like we indicated, so 2 alleles. And we'll get into things where you got multiple alleles flying around. And that's okay. We have our way of handling that. But when you get into something complex like us, it makes sense that there are many ways to basically execute a gene. Okay. Oh, wait a sec. This says 25. Take that. All right. Okay. So let's look at a bit of a situation here. Each time that a zygote is formed, so let's go back to zygote. Now, zygote's going to go on in mitosis, but it's the result, it's the end result to meiosis. So again, that's why I like it as a jumping point. In a chromosome, if you lose a big giant region right here, see that? If you lose this region, it looks small right there. Let's say this is chromosome. So chromosomes go in size. 1 is the biggest, 22 is the smallest. So if, let's say, you lose a small piece of one of the smaller chromosomes like 15, 16, or 17, this region is still, what, hundreds, maybe thousands of genes? Just gone. Totally gone. So if that forms in a zygote, that cell is going to have an atypical set of instructions. The book has had pages ripped out. Now, another key thing is that you may say, well, how many traits can 100 to 1,000 genes control? This isn't quite on the exam yet, but remember, most genes have a certain amount of partner genes with them, and those separations of degrees can span a ton, right? So these 100 genes may actually affect 10,000 genes, at least slightly. So even a small bit of info missing is going to have pretty big consequences. Okay. So given that we have the genome, given our knowledge of mitosis, given what we know is important about the genome, kind of bringing up a point here. Every time you make an advance, the genome is an advance. Every time you make an advance, you bring a consequence with it. Agriculture brought the spread of disease. Domesticated animals are incredible harbors for some of the diseases today that have killed millions of people. So the beginning of smallpox, measles, anthrax, cholera, lots of stuff, malaria. Similarly, could any of us do our day-to-day without a car, right? Engineering, it's a marvel, right? Everybody in this room likely knows of somebody who's been affected by a car wreck, right? And obviously the last one, the internet. Freedom, information, everything, Wikipedia at your fingertips. But unfortunately, as we all can imagine, not the most fun or nice place on the planet. Okay. Yeah, you knew I'd come here, right? So, one thing that we have an advantage with right now is IVF, in vitro fertilization, okay? The idea that with a couple that has a hard time conceiving pregnancy naturally, you can take eggs from the woman, sperm from the man, and try and, in the lab, create the beginnings of a zygote and then an embryo. So right now, a successful IVF can be, you get maybe 4 to 5 embryos. Good enough, right? Now, it's no problem to go in and take one of these cells, because they'll just replicate another one anyway, and sequence it. Say, what's the genome? So you have a choice suddenly, right? It's like, what if the mom says, oh, no, we only want girls? Or the mom says, no, we only want boys. Choose that embryo. Now, IVF's been popular, or sorry, not popular, but able to use for 20, 30, maybe more years. Everybody's all excited about CRISPR and stuff. We've been able to do this for a long time already. Now, the video that I had kind of posted, the guy kind of uses the IVF as an example here too. Let me just scroll for you back to the response here. And you can just fill this in as we go. Imagine a scenario where you can make 10,000 embryos easily, right? And of those 10,000, you have such an enormous number, you can pick and choose which embryo is the perfect set of traits you want, right? You don't want an embryo that's gonna have kidney disease, right? No, we don't want that one. You don't want an embryo that maybe they're gonna be kind of too short or something, I don't know. Polygenic type cosmetic traits, right? Now, IVF is currently still available to use and make that call. There's one choice in here. It is a bit of a strange situation. When IVF was first starting, there was a case of 2 parents that had the trait for dwarfism. And they indicated strongly that they would like to choose an embryo with dwarfism as well. Now, the doctor advised them there are certain health conditions that come along with that. Is that fair to the child for you to choose that fate right there? But at the end of the day, too, is it not positive to have sort of a piece of you, kind of help you who you are? And that's the quote that comes up with IVF a lot, where it's like you've chosen this child's fate and what they're going to be. And a lot of the times when you speak with people with atypical traits, they will say that my trait made me who I am, and I rose from that, and I feel empowered by that, and I'm really glad that I was, that I'm here. So the fact that we have this choice, and the fact that this is widespread to a degree, we're already sort of knocking on the doors of this genetics question. That power, that revolution, we've had this for quite a while. We don't talk about it quite as much as we can. Now, sequencing and IVF is only going to get better, not to say that it's not extremely expensive. Okay. So you guys are being pretty cautious. Last year, I had a couple people be like, nah, kick the door down. Let's sequence, let's make super babies, right? I had a lot of people do that last year. I was like, dang. Because remember, science doesn't have morals to it. You have to combine both. You have to be somebody who can do both. You have to, because you can't measure humanity. You can't measure justice. in this system, right? Those are intangible, intersubjective human things. So having somebody that can do both and think through both, that's what I want people to come out of this class with. Okay, I'll leave that up for a little bit. Now, consider the answer that I gave you up there, but consider this just, again, a little practice writing. You don't have to write anything if you don't want. But think about why you selected your answer. What was the piece that was most important to you? And that quote I think I shared yesterday too, control the gene, control the future. That comes from before we had the tech to do IVF, when people did want to do that. Can you imagine what this technology would have been like in the hands of somebody like Goebbels, one of the Nazi propagandists that was a big eugenics person? We will examine the pseudoscience of eugenics probably next week from now, I'd say. So there's a lot of factors and information to consider. One of which, and you'll keep seeing this in biology and life science technology, it's expensive. It's not free. We'll see that with certain cancer therapies too. Even the lymphoma I work on is something like, there's a treatment that costs about $250,000. Science sometimes goes so quickly that that's not accounted for as well. Last point I added in just today, which was, because I had read an article about it. Is it ethical to go and advertise like, hey, can you donate, would you donate an egg? Would you donate sperm for money? Would you help this IVF couple? But we'll also pay you $500,000, let's say. So on any free write type stuff, you can always like find me for feedback, like would this score points if this was out of 5? And I can give you like strengths, weaknesses, top 3, things like that. Because I do like having targeted writings there to kind of help you kind of improve that way. So always an interesting question for me from my perspective, just because it would be a strange conversation to have with your parents to be like, so actually, you were the opposite of a mistake. We actually chose you out of 1,000 different embryos. So go clean your room, like be appreciative that we chose you. Be bad, sorry, a little dark. Okay, so given IVF, and obviously where a lot of heads will go towards this is Roe v. Wade, which is where's that line cross as far as cells to human? There's one that I wanted to introduce a little differently though, Park v. Chesson. Given IVF's ability to sort of predict the future about a child, let's say, at least with a certain degree of certainty, a family, Stephen and Hetty Park, went to a doctor, Herbert Chesson. They said, we want you to screen and make sure our child that we're about to IVF does not have any atypical traits, specifically X, Y, and Z. Chesson didn't do the screen right, and he didn't inform them that their child's karyotype did have a deletion at a certain region. They carried the pregnancy. They had an atypical trait. They sued Dr. Chesson, and they won. So by precedent, wrongful life is a legal precedent. It's a strange thing to think about. That in the United States court system, you are on the right side of the legal interpretation of this biology case, that certain lives, if chosen by the parents, are wrongful to come to term without their knowledge. It's negligence on the part of the doctor. So again, you don't see biology in the legal system very often. You will start seeing it a lot more with genomics, with cancer therapies that are, the way that they're getting more involved, let's call it. And so being prepared, seeing that V for a lot of stuff, it'll start to help you think about it. Okay. So again, not really talking about embryos at this point, more talking about a cell versus all the pieces of a cell. Because the cell is a living thing. We can't really argue that. It makes its own energy, can make copies of itself, stores information, hooray. All the pieces of a cell, not alive though. Plasma membranes, that's not alive. DNA, it's not alive. Mitochondria, yeah, we can argue. But all the pieces, only when they come together can they form that strange union that we study and call life in biology. So as we go through mitosis today, sort of remember that even when we're considering human cells, because I will end on cancer, these are very much living things by our definition, even if they are part of a greater whole. Okay, so on that, you can consider this blue or green text. Sorry, I didn't have a title in there. But you got a lot of cells. You got a lot of organs that make new cells. So kind of gross, but think about your digestive system. A lot of acid, a lot of stuff flying off, a lot of dead cells just shedding off that. You need to renew your digestive system cells all the time. You are constantly renewing your skin cells as well. They're constantly like falling off, right? There's plenty of cells in your body that do not renew themselves very often. Neurons are a good example. Your heart cells are an even better example. The heart you're born with that is beating today, rarely do those cells ever make mitosis copies of themselves. It's just going from the time that you came out, basically. Obviously, you had to form the final thing. But now, given that all of these have the same genome, just like we talked about. Every muscle cell, every heart cell, every neuron, every kidney cell, they have the same set of instructions. It's how you use the instructions that matters, though. So this is a GIF or GIF if you're wrong. You can go and hyperlink this if you want and watch the video. It's really cool. I always like to do this. Who says GIF? And then who says GIF? Oh, it's okay. We can agree to disagree, right? Okay. Last couple bits here, and then we'll be on to our break. So, had to come up against the hardcore stuff eventually. Here we are at the cell cycle, and we've seen this before. What I want to do is a teaching technique called selection. I will find pieces of this that I find more important to the outcomes of this class. I will try to indicate that those will be what I test you on. Anything in red technically is going to be that fair game. Okay. First thing is that to remember, mitosis is a very small part of a cell's lifetime. And like I said, lots of cells don't really do mitosis a lot. They're fine just hanging out. So the cell cycle, mitosis is a part of the cell cycle. It's a small part. Only this amount right here, let's say. Most of the time, and most of your cells, they're just going to stay in that little G0 right here. Okay, if we can see it, there we go. G0 just means they're doing their job, hanging out. They don't need to divide. They don't need to die. They're just doing okay. So honest to God, you spend most of your cells spend a lot of time here. Like let's put it conservatively at 90% of your cells are just hanging out most of the time. Now, should the time come to renew cells, or let's say that we are in the intestine system and we need to make lots of copies because the intestine system is kind of difficult and it's going to kill you. So you got to make more copies of cells. Let's start. G1, easy stuff. We're going to start mainly duplicating all the nutrients, mitochondria, things like that. So I'm going to ask, oh, sorry. Are there enough nutrients to divide? That's what G1's main role is. Do we have enough proteins, carbs, nucleic acids for DNA, and fats, phospholipids? Basically, are we healthy enough to even do this? And moving on, if that's okay, and we feel good, and we do need another copy of this cell, we're going to go from 1 cell to 2 cells. We take a very important and not a light step, but S phase. This is the duplication of DNA. You start with 46 total chromosomes. You will duplicate exact copies of each of those. So don't break the math down here if it's easier. Remember that You've got 23 from mom, you've got 23 from dad. That's 46. You're going to be doubling that into exact copies. This is where the word chromatid comes in instead of homologous chromosome. I'm not going to strictly test you on that, but given that you have the notes, I'll probably expect you to distinguish the two if I ask about it in a question. Chromosome, let's just for an example, chromosome 6 from mom and chromosome 6 from dad. Those are homologous chromosomes. There's a human chromosome 6 and a human chromosome 6, but your mom and dad are different genetics slightly, 99% alike. But in S phase, you are 100% just copying both of those two so that you have a copy to give both of your cells. So you go from 1 mom and dad each to 2. So S phase for me, for this class, because this is a DNA, this is a genetics class, that's the important step, I'd say. That's the important one that I'll likely ask about or sort of test you on more often. Okay, so zooming in. G2, think of G2 as the final check. You know how you get in a roller coaster and the guy checks and make sure your seatbelt's on before you fly out or something? I hate roller coasters, by the way, so it's kind of what I'm always scared of. This is that final check. Are we good to go? Are you strapped in? Do we have double DNA? Do we have double nutrients? Do we have any damage to this cell before I make 2 copies? I may as well just annotate that when I'm talking, sorry. So, times 2 DNA, times 2 nutrients. And is there any major damage that we detected? Because you do not want to set up to make a copy of a bad cell, right? So G2 is the final say okay, we're a healthy cell, let's do this. Because if one cell's bad and you make 2 of them, now you're obviously causing a problem. Okay, so that's cell cycle. Everything that we did here is to get to mitosis. So I think something that helped me in undergrad is separating those 2 processes. Mitosis is a small part of what we just learned. It has the details that we need to just start detecting damage when chromosome stuff happens. So that's why we teach it, and that's why I also like to bring in cancer with it. But again, it is still a very small portion of the major lifetime of your cell. Okay. So let's focus on S phase a little bit. I like this image because it just plainly shows what's going to happen here. This could be any one of your chromosomes. Let's just say this is chromosome 7 for mom. So you're doing this 46 times. All you're going to have during S phase is that chromosome duplication. You had 1 up here, and now you have 2. And remember that these are sister chromatids. These are the exact same chromosome. They're just little clones. And like I wrote down and scrawled there, it's because you are making copies of the cell. You need to have the exact same instructions to the next cell. You really do. And we're not quite here yet, but you can imagine the damage that I talked about if you lose a certain portion of a cell, right? So if during S phase something goes wrong, and you lose this chunk right here, right? That would be bad. So that's why this process needs to be maintained with a bunch of integrity. And that's why I'm hopefully, kind of, sorry, monotone going through something you've probably seen a few times. But if we want to get into hardcore DNA damage, this is one of the places that it can happen. But usually you make two nice copies, and that's great. Don't worry, we're almost at the break. Okay, now this isn't necessarily content. This is another study strategy that I learned from med school that I was trained at. So I didn't go to a normal PhD granting university. I went to a medical center. So the people that taught me how to teach were medical professionals, doctors, nurses, allied health professionals, dentists, anybody, name it, all in Omaha. Matrices are one of the best ways to study large connected processes that, as you can tell, there's different phases, but there are specific things that you can sort of hone in on for each phase. Matrices also make this a lot easier to learn. So imagine just taking linear notes on each one of these things just in a page, right? Not quite connected. This makes it a little easier to compare across and see the differences and similarities between each stage. To me, and to a lot of medical type students or allied health students, that really helps. Now, the place this really shows up later is if you take like pathology and it's like disease, like all these diseases you got to know on the top. And then you can say on the left there, like symptoms, treatments, causes, et cetera, like each little factor. So this is sort of a way to study. Disclaimer, it obviously doesn't work for everybody, but I always do think that it can kind of help your notes become a little more efficient. And I think this is shared too, so you can always like print stuff off like this. All right. Here's mitosis. And I'll be honest, these are probably the only steps that I need you to know. And I'm about to scroll up to each one because I know these slides are not shaped enough well. Okay. What is happening to the DNA in each stage of mitosis? That's the main question that I'll probably ask on any exam. So if we're looking at prophase, we're sort of assembling the DNA. As you can tell, it's kind of in a big mishmash. It's dyed blue in here. The DNA is assembling, it's getting collected. We're making sure that an even amount of genetic material is going to go to cell number 1 and cell number 2. Going to make sure that we're making this a clean swap. This isn't cell bio, but there are some fun details about how everything evens out nicely. Prophases, though, this is kind of your like, if you got a crowd of people, they're kind of going towards this way and that way if they're meant to go to different ways. All right. Metaphase, I like this one because it's usually pretty easy and it's like the pretty picture that you always see. This is where the DNA is all the way aligned. It's all lined up, 1, 2, 3, 4, 5, 6, 7, all the way down to 46. So you line up all 46 chromosomes because remember, exact copies are going to be aligned like this so that when they split, copy number 1, cell number 1, copy number 2, cell number 2, all 46 times. So metaphase is that final check. Do we have everybody? Are we ready to ride? And then anaphase is what it looks like. Split. Let's do this. Rip it apart, make 1 cell into 2 cells. This is the most forceful part of the process. This is where the most mistakes can ultimately happen, sadly. Sometimes that rip can bring over maybe 2 copies accidentally, right? Now, typically your cells are pretty good at this though. So you usually don't make any mistakes. Then telophase. Think of this, we're kind of at the end here. I would call this like sort of reassembling 2 new cells. Because you can kind of see in telophase, it's the only one where it's like, now I can see 2 cells at this point. That's about it. one way I do test on this is I can use pictures sometimes and say, again, there's not too many super creative ways to test on this to meet the outcome. And that's why hopefully your little note cards can take care of you on this one. We'll get into some greater details when we get into big, massive structural damage that can affect this. But for now, the basic process is really good and important. Okay. So last little thing. Then I'm actually going to give you guys the break because I've been talking for too long. So blue text, y'all know how bacteria evolve, or sorry, they do their mitosis is something called binary fission. It's a very somewhat similar process. We're very different cells, but they're just two clean copies. The roots of mitosis exist in prokaryotic division, but we really don't have the tools or inference to really go back and look at exactly what the first cell did to make one cell turn into two cells. We know that at one point, because everything universal in life does follow these cell mechanisms, that at one point back in whenever day it was or whatever, one cell existed for a moment, just one, and everything came from that one thing. Kind of fun. Okay, last little bit before we take a break. I promise, I know I said that like twice. So, fun little paradox here. This ship called the Theseus comes into the Athens port like, 2,500 years ago in Greece. It was like a big warship, hooray, it was a big monument. Now, the ship gets old as it sits there as kind of a museum piece in the harbor. Salt water's kind of angry, rips it apart. You start replacing pieces of the ship over the years. Maybe the mast in 5 years, maybe the deck boards in 10 years, and in 20 years, the whole thing's been replaced. It's not the same ship anymore. Same interesting thing that happens to us. Nearly 100% of your cells are different than they were 5 years ago. And the interesting thing with that is, and we'll get into aging and mortality and everything, they're about 5 years accumulated with damage. Aging is something that's very interesting in biology, and it's something we'll get into a little more. But mitosis is a very good process for keeping up with things and maintaining who and what you are despite that. Okay, hooray, break time. Take about a minute. That was a lot. It's a lot of cells. Now's a good time if you got questions, anything. And now's an excellent time, if that poll everywhere didn't work, to come find me and get a little piece of paper. Hooray. Just raise your hand if you need something, or just come and grab it. Yeah, just in case, yeah. And if you feel like just in case the poll everywhere might not have worked, you can always just do a backup plan. Yeah.
So where do you look to see
like what the questions were? Just kind of write like a more general. You can write like kind of your own general answer, and that's okay. That was the IVF question. You're on mute. Scout cat. Enjoying. All right, let's do. Yeah. All right. Cell cycle time, the end. We'll kind of end on some tumor stuff, but let's end on some final red text. So back to cell cycle, not mitosis. I kind of covered these, right? It kind of was when I was talking about G1, G2, and all that. Here are the needs that you have in G1. Basically, like, are we healthy? Should we do this? So these checkpoints, these are the reason why it's a cell cycle. Because a lot of the times, if we're not healthy here, the cell actually undergoes something called apoptosis, and it blows up. You got 10 trillion cells. Getting rid of one bad one is not going to hurt you. It's actually going to be way better. Okay, G2. This is sort of the finale checkpoint. This is much more involved. This makes sure you have all 46 times 2 doubled. And this is going to make sure for the final time that you are not sending off damaged cells. You do not want to copy damaged goods into 2. So G2, yeah, I'd say it's a little more stringent, and the cell can blow itself up at this point if it feels unhealthy. Okay, last point, spindle. This one kind of makes sense, and this one actually is in mitosis. So remember in metaphase, oh, it's written there, sorry, again. In metaphase, when the DNA lines up on itself, this is the final check to make sure everybody's here from the party, we can go. You make sure that these little connections are all made at every chromosome. Because as you can imagine, if you just leave something loosely sitting there, a piece of DNA, it's not going to find an even home. So 3 checkpoints. It's the makings of a multiple exam question of, oh no, this is happening. What checkpoint is going to trigger the cell to undergo apoptosis? Okay. Now, don't worry as you see all this flying around, go versus stop. This is the next part of the equation, which is as the cell decides, this is the makings of the decision for does the cell copy itself or not? Go versus stop, to put it plainly. In red, you see a lot of stop signals. In green, you see a lot of goes. It is the accumulation on one side or the other, stop or go, that makes that choice. Eventually, if a cell needs to go mitosis, the go signals start to rise up. Now, it's totally okay to just generalize the stop signals as stop signals. I will never ask you what is, ATM, what does ATM do, right? This isn't cell bio. I want the general idea that you have a wealth of stop versus go signals, and that decision is made by a numerical decision. Okay. Your typical go signal is something called these, though. These cyclins and CDKs. And these are just down here. So again, I'm going to blue text phosphorylation a little bit in there. It's good to know why they're doing this, but it's not what I'm testing on. I'm testing on what does a go signal do. So a CDK is a big gene. It's a protein. Now, biologists, we can just draw it as a big cartoon like we have right over here. In the event that the cell receives enough go signal to start mitosis and start this process, a cyclin, another gene, and a CDK will find one another, and they will combine into sort of a mothership that will start to say, go, 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 go. Sort of like an avalanche, right? One piece pushes a bit of snow, and that snow pushes more snow, and all of a sudden the mountain's coming down. It's like a cascade. I'll leave you with this red text right here. In terms of a go signal that I want you to feel comfortable with, those two words are the main goes. They are the signals that accumulate and say it is time to double, it is time for mitosis, we need more cells. So a great example of when you need more cells is wound healing. At your wound, you need to repair that, right? You need more copies of skin cells to seal that up, muscle, all that tissue. In that area, you will have a ton of go signals. You have a ton of cyclins and CDKs. This is their full name down here, sorry, cyclin-dependent kinases. Let's just do this. There we go, sorry. Okay, so CDKs and cyclins are the sort of mounting signal. Piece by piece, they get more and more and more of them get recruited. Consider this step one towards that decision to do mitosis as a go signal. Their role is to ultimately initiate the final go signal, something called MPF, mitosis promoting factor. So the entire role of cyclin CDKs is to unlock the one thing that will trigger mitosis across the cell. It's like a master key, MPF. Very powerful. But a lot of keys have to get unlocked. You ever see the movies when they're about to launch the missile and like multiple keys have to go in the thing and hit the button at the same time? It's kind of what's happening here. This decision is not made lightly, and biochemically it takes a long time. So cyclin CDKs are going to combine. Ultimately, they are going to trigger MPF to show up. And really all you got to need to know about MPF is it will trigger mitosis. Experimentally, every time. Now what I mean by that, and the next slide is sort of to show you how powerful MPF is as far as a go signal. If you just add MPF to a cell that's just minding its own business, the cell starts to freak out and undergo mitosis just immediately. So that's why MPF is kept under all these signals, is because you don't want mitosis to happen unless you really, really need it to. Okay. So kind of talk about this at this lecture because it's fun, because mitosis can be somewhat of a boring process. So sometimes I try to get into it a little bit more about where I come from. If I was a fly person, I talk about flies. If I was a plant person, I talk about plant person. But I'm a cancer person, so I talk about cancer. What if go signals take over? I'm showing you a blue text example of something called EGFR. The GF is growth factor receptor. This is a signal to go. It's one of those go signals, kind of an off-brand go signal. Sometimes a mutation happens that causes this signal to stay on permanently. Constant flows of go signal. Now, one of the other consequences that you see, proliferation is another word for spread, multiplication, mitosis, tons. So if you have an on signal that is stuck, the cell will likely not know what to do with that. Cells are not typically smart things. If they get a go signal accidentally, they'll keep going. They'll keep mitosing. The other thing is down here. It's a piece that I will kind of expect you to elaborate on once in a while, which is cells also in the mitosis process typically are triggered to survive. What I mean by that is, remember that decision to blow up at the checkpoint? Mitosis can override that if the signal is high enough. So the combination you're looking at and where we're going with this is going to be cancer. Two pieces are needed for any cancer, two basic pieces. You need a lot of cells, and they need to survive. Sorry, that's out of order. That bothers some people. Good example why you need both factors is everybody's seen a mole on their skin, right? Maybe. A mole is just a bunch of cells that did too much proliferation. Oops, but they died off. They didn't have the extra gear to survive. And specifically, you run into stuff like this. A stop signal, BRCA. This is a very excellent and powerful gene, and what it does is it scans your genome and it scans your cell for damage. I'm writing this little note up here. It scans during G2. I showed you that checkpoint, and now I'm showing you why it's a checkpoint and what actually tells that decision, what actually informs that decision. This is one of the ways. BRCA is excellent at finding these damages. Let's say there's a break in the DNA. It finds that damage. Oops, I should have done red. And it triggers a cascade. Now, the cool thing about BRCA, it actually tries to repair the gene first. It actually gives it one shot to repair. But if the damage is pretty severe, like we see in this double strand break, a lot of the times we're going to head over here. We're just going to blow up, call it good, we're done. We don't want this to turn into cancer, just get rid of it. So a stop gene not only kind of tamps down the signal of, hey, let's do mitosis, it also checks to make sure your cell integrity is okay. That's that second role of a stop gene. We'll get into this when we get into Mendelian. And some of you are probably already familiar with BRCA's role. But you can imagine that if you're only born with one copy, let's say it's from your mom, and your dad's copy is defunct and doesn't work, you better not lose that one copy, right? Okay. Second stop protein. I would call this, again, a kind of a blue text. P53. So BRCA, it was very good at what it did. It's a good stop protein. P53 is the ultimate check. It is the ultimate checkpoint gene. It checks your entire genome, your cell, for everything. And if it detects even a single wrong thing, it goes nuts and blows the cell up, prevents cancer. Because it can detect when there's DNA damage. What that does is it activates P53, and everybody stops at G1. See it? There it is. Again, it tries to repair it. But if you want to be safe, may as well just kill off that cell. Like I said, you got 10 trillion cells. It's okay to get rid of a couple if it seems like they're damaged. So we call this is true green text. We call this the angel gene because it is by far our best protector against sending off damaged cells. And that's why when you have a non-functional P53, all the damage in the world goes unchecked. There's very little stopping those checkpoints, and damage can accumulate and get worse and worse over time. People that are born with two non-functional P53s typically develop a variety of cancers because every time their cell divides and there's a mistake, probably don't catch it. It's a very tough condition. Very rare though. Okay. Last little 15 minutes here. This is where we've been going. That imbalance of a stop and go, all that is, that's a tumor. Now, a tumor is not going to show up from one change, one mutation. Now, mutations can either knock out stop signals or they can enhance go signals, right? That first mutation, my genetics prof kind of indicated this. It's like, if mutations are these 1 in 100 million things, how does cancer ever happen? And the key is that the first mutation is typically very subtle. This first one right here, it's just slightly faster growing, just slightly. But if something's slightly faster growing than all its little cousins in its neighborhood, it'll outcompete all of them. And suddenly that region is comprised all of cells with that one subtle mutation. So now that you have 100 million cells that have that one mutation, making another 1 in 100 million mistake is not mathematically out of the question anymore, right? Mutation number 2. So let's say this was a go signal. Now you knock out a stop signal. Now suddenly this clone right here, a little more aggressive, a little angrier. Say in one of these, because remember, this is going to grow and this is going to go times, like 10 million, it's a lot of cells, which means there's a lot more opportunities to make more bad copies somewhere in that math, right? So you get a third change. And as you can see, kind of from this little graphic right here, this is indicating that we are now completely out of control and we're growing like crazy. And let's say to make things worse, these don't just grow, they survive. And this is how mitosis can get co-opted and the process can just run out of control. Cancer is a pretty common thing in our lifetime. 1 out of every 2 men in this room will encounter some sort of cancer diagnosis. 1 out of every 3 women in this room will encounter some sort of cancer diagnosis. It wouldn't be hard to imagine that everybody in this room has somebody in their close to immediate family that has cancer. It's a universal feature of life with mitosis, but it's not a perfect process. And as you get to this aggressive stage, the population starts to evolve like an animal population does, with different variations. Some are stronger, some are weaker, but some can avoid the immune system better, some make more energy. We'll get, one of the final lectures of this class will be when we've learned everything, we revisit cancer, and I'll show you why it's so hard to treat. Now, as far as defining these stages, we don't really have concrete definitions, but we've all heard the word benign before. You can have a benign growth of cells, maybe an uneven, splotch of skin after a sunburn. Yeah, they grew a little fast, but they didn't survive. It's okay. Now, malignancy, that's cancer. Malignancy equals cancer. This group and population of cells is growing without stopping. They're not dying in response to your body's signals to tell them to die. And mitosis is going to keep going. Just in a complete cycle with disregard for the normal signals. There are 2 other names for this stage. What happened to our go and stop genes? You can now call these oncogenes. You can now call stop signals tumor suppressors if you want, now that we're in this stage of what we're looking at with out of control mitosis. Okay. That's your base look at what a tumor looks like, but there's one final stage, and that's metastatic cancer or metastasis. This is where, should you have a tumor on your liver and it gets so out of control that pieces of that 100 million tumor cell or cells, there's tons of them, maybe a couple of those cells can break away from the tumor, enter your bloodstream, and they float around a bit, and maybe they find a spot to latch and start growing. Because your liver tissue doesn't look a lot like your lung tissue or your bone tissue. But to the tumor, sometimes they can evolve so aggressively that they can become fit to survive on a new tissue. Basically like us going to Mars and colonizing it. That's a sad example for humanity, sorry. I don't think we're a cancer. Okay. Because the thing you have to consider with a tumor is that it is a population. It is not a clone of itself. You have a swirling amount of genetics and evolution happening in these things. Every patient's tumor is different. So up here, like we talked about, this is that first mutation, something we call a founder. So this is blue text, so I'm not going to crush you on the test with this. This is just interesting. Now the founder will branch into populations, kind of how animals and species branch. And sometimes those changes can make something a little more aggressive. Now the cool thing is that a lot of our medicines are great at eliminating here. We can even sometimes eliminate some metastatic cells, right? Our medicines are pretty good at treating the initial cancer. But even if you eliminate all these, all our medicines go after stuff that is doing mitosis a lot. Problem is that you can't kill this one very easily because this one looks a lot like your normal cells. So even though you killed all this up here, we can just start one more time and evolve again into a new pathway, a new metastasis, and we're back. Finding that quote unquote cancer stem cell is an area of research we are expanding, but it's difficult because they look very close to what a normal cell looks like. And you can't treat them with much of anything because it'll kill all your normal cells, right? Okay. You don't talk about, and a lot of you going into medicine, so it's a little grisly, but metastasis, like I said, this is seed and soil hypothesis. Liver, a pancreas tumor may find that it exists well in bone. And one of the things that happens with metastatic tumors is that they're big enough and hard enough that they can break legs and stuff. That's how nasty this gets. Most people die from metastasis because the lung is a very good spot for tumor cells to find themselves. They expand, you can't breathe anymore. Same with the brain. Any space that's taken up by that is going to be a bad deal. We'll address this question later with some genetics that we have now. But what you're looking at, SC9A, and in all its biochemical glory, is one of the major human pain receptors. So given that, if somebody's in hospice and you're in immense pain with metastasis, consider what lengths you'd go to alleviate that pain for that person. Let's say the treatment isn't an option anymore, it's just eliminating this. Keep that in the back of your head because we will see how that's possible maybe in this class. Okay, and this is just a thought question too. I'm not downplaying COVID, but we're pretty good at treating it when it happens and, you know, figuring stuff out. You can't vaccinate against cancer because everybody's got a different role of it and cancers evolve differently. COVID's simple, stupid, but very effective. Cancer always can change, shift, react in ways that other diseases can't. Because remember, they're not they're living things, they're a living population, but they're technically you. So one thing that we use is chemo. Remember mitosis? Cancer cells need a lot of mitosis. That means you're dividing the DNA all the time. Let's send in a chemical that disrupts DNA replication. But that's why chemo is kind of like carpet bombing, okay? You're just gonna try and annihilate everything and it's just gonna kill the cancer faster than it kills the patient. Nothing wrong with chemo. You can cure a lot of cancers with chemo, but it's not targeted. So all this slide is saying, you don't need to know each of these pieces. It's just saying that mitosis represents our best target to kill cancers because cancers need mitosis, unlike the rest of our cells as much. And in the green text, if you ever want to really sound smart, you can use the word synthetic lethality, a strength of something that exposes a key weakness. Strength of the tumor is that it can grow like crazy and escape a lot of stuff. The weakness is that it needs mitosis all the time. And if you limit it, you can kill it. Uh-oh. There you go. Okay. Problem is, and don't worry, we're not doing all slides. Problem is that cancer can evolve in a myriad of ways. It's a very, very cool system to understand evolution from. It can be progressive, it can branch. And remember, in a single tumor, you probably have different genetic populations that are emerging. And that's why it's really tough to use a single therapy to kill one population. So if you guys ever want to talk about even, yeah, some of the animal people in here, if you ever want to talk about evolution, I'm a big person on it. Now we're trying to use massive sets of data to find patient populations, for example, that I've got right here, where their tumors behave sort of the same because their tumors sort of went one pathway in evolution. That means that we can maybe start targeting drugs to these people specifically based on their genetics. But sequencing right now, and as far as predicting cancer, it'd be pretty tough. Small text up there outlines, you know, the raw solution. Sequence everybody all the time and at frequent intervals, but that's not feasible. Even that, you'd have to sequence your liver, your brain, your lungs to just find any DNA changes, right? Now, this is blue text, it could be green text, but something called circulating tumor DNA may offer an effective means. We are finding that when tumors, even before they're detectable symptom-wise, they shed pieces of DNA because they blow up because they're dividing so fast. Plenty of tumor cells just like blow up and die because they're like basically stuffed in with the other cells. We're finding that we can isolate this DNA before a patient shows symptoms and say, oh boy, let's take a look here. And we can catch it early. This is a big deal with stuff like pancreatic cancer that's really hard to find. Okay, don't panic. But let's write again. Points on that are not exam points are always fun, right? Let's do a little exercise. Let's make this do. You've always wanted to boo a professor, right? Boo if you want me to push it later. See, tomorrow at midnight. Boo. Yeah, boo. Otherwise, it's going to be due tomorrow at midnight. Oh, you guys want? No, I was actually going to push it till next Thursday. You could have gotten me so much farther. Okay. Let's do this. We got the time. I want you to come up. Yeah. Question or experience you have on cancer. Like I said, I'm a cancer person. I like addressing these. I share the examples of the class. Let's do this next Thursday at midnight. Does that sound cool? That gives everybody time. You can do it today if you want, but that gives everybody time. What I'm looking for, think of a question. This is a targeted writing practice at asking a question worth asking. Think of the question that you're going to ask. It doesn't have to win the Nobel Prize. It can be small too. Think of it that, would somebody click that question to find the answer? And equally, don't find something that's a yes or no off of Google. Remember, think of me as your audience here. I can clarify a lot of stuff. It's very fun. If I was a plant person, we'd do this with plants, but I'm not. These are some fun examples up here. Hopefully we can read those. Maybe not. Okay. So I might, yeah, why don't I post some of these slides? Now remember, don't just copy one of the examples. I'll definitely give you the 0 and make you revise then. But essentially, it's sort of like an in-depth way of looking, okay, we looked at the simple way that these happen with mitosis, but I've always had this question, like a common one that always shows up is like, what percentage of cancers are environmental versus what percentage are hereditary? It's not a bad question, right? I kind of oversimplified it now and used the example so you can't, but that's kind of what I'm looking for. It can be broad, it can be specific. Go for it. Next Thursday at midnight, we'll go from there. All right, have a good weekend, everybody.